Hi everyone, let's get started and make it quick and easy peasy. Now, first, let me explain the history and introduction of microbiology. These topic does not carry much weightage in the exam, but they often come as 3 marks question. So started with Antony Fils one Louis Van Hoek from 1632 till 1723. He was known as the father of bacteriology. He observed bacteria and other microorganisms in single cell microscope which is constructed by him. So he named those small organisms as a little animalcules. He also described the different shapes of bacteria such as coccal, bacillary and spiral. So these basic shapes of bacteria we will discuss in detail in next lecture. Next is Edward Jenner from 1749 till 1823. He is famous for introducing the modern method of vaccination to prevent smallpox virus, which was one of the deadliest disease at that time. The fun fact is that the word vaccine and vaccination were later popularized by Louis Pasteur as a tribute to Jenner's groundbreaking work. He used the cowpox virus, also called the variola vaccine, to immunize children against smallpox virus. From these, the term vaccine was actually derived. Jenner did not just stop there, he made many other important contributions to medicine. He was probably the first to link angina with hardening of arteries. He also described the rheumatic heart disease and even purified several important medicines. Now next is Louis Pasteur from 1822 till 1895. He is also known as the father of microbiology. He was originally trained as a chemist, but his studies on fermentation led him to take interest in microorganisms. His discoveries completely changed the way we understand microorganism disease and food safety. He gave the principle of fermentation for food preservation. He introduced the sterilization technique which became the foundation of safe laboratory practices. He shows that some microorganisms are not destroyed by simple boiling, so for sterilizing fluid, he recommended heating them to 120 degrees Celsius under pressure. For sterilizing glassware, he advised using dry heat at 170 degrees Celsius. He also demonstrated the value of cotton wool plug, which protects culture material from aerial contamination. He even coined the word vaccine in honor of Edward Jenner cowpox virus. Between 1860 and 1861, he disapproved the old theory of spontaneous generation, which claimed that life could arise from non-living matters. He was also the first to culture staphylococci in liquid medium and even produce abscesses by inoculating them into rabbits to study infections. In 1881, he developed a live attenuated anthrax vaccine, which is major step forward in immunization. He also devised the process of destroying harmful bacteria in food and drink. This process is known as a pasteurization and it's still used today to make milk safe to drink. Next guys, meet Robert Koch, one of the legend of microbiology. He gave us method to isolate bacteria in pure culture and he invented the hot air oven and even explained the hanging drop method to check motility. He also discovered the solidified agent and introducing them staining techniques using aniline dyes. Now let's talk about the Koch postulates the golden rules to prove that a microorganism can cause a disease. Rule number 1, the organism must be present in every case of the disease. Rule number 2, it should be isolated in pure culture from the lesion. Rule number 3, when these pure culture is introducing into healthy animal, it should produce the same disease. Rule number 4, the organism should again be re-isolated from these experimentally infected animals. He made golden rules to link germs with disease, but guess what? Yeah, not every germ follows the rules. Some bacteria are totally rule breaker, like behind leprosy, syphilis. For example, Treponema pallidum just refused to grow in the lab. See, even germs love to break the rules. Finally, last one guys, meet Polylech from 1852 till 1915. He is the father of chemotherapy. He was the first to report the acid-fall nature of the TB and also developed the techniques to stain tissue and blood cells. He also proposed the toxin-antitoxin interaction which we now call Illich phenomena, and he also gave the side chain theory to explain antibody production. He gave us the idea of magic bullet, which is a drug that targets the germ without harming the body. His discovery Salvarson cracked syphilis treatment wide, and that idea it became the bear of modern medicine. So that's the end of the today video guys. But before we wrap up, here are some super important questions. If you found this helpful, don't forget to subscribe, like, comment, and share with your friends.